Okay, so let's proceed with today's lecture. Okay, so by the end of this lecture, in terms of expected outcome, okay, I'll be expecting that students should have a very firm understanding on B-cell action and differentiation. B-cell action, activation, and differentiation. We have already covered the canon fodder of this, okay? Also, the concept of immunological memory and antibody structure significance action. In your introductory basic immunology classes, we did tackle aspects of antibodies, types of antibodies, and their structure. And now we will add to that and try to see how best we are able to connect it all in terms of the B cell itself, the immunological memory, and the significance of these antibody structures that we have described previously. And what is important is that we are trying to lay a ground for us to have a comprehensive understanding of how the immune system is interrelated. I want us to reach a point where we should be able to just think of the immune system and be able to describe what happens when an antigen enters or breaks the immune defense system, the primary immune defense system. What happens when an antigen breaks the skin and has access to the immune system. We want us to be able to describe what happens from then on, how the innate immune system engages that particular antigen. If the innate immune system is not able to handle it, how is the adaptive immune system engaged? What cells of the innate immune system are being activated? How are the neutrophils, the macrophages, what cytokines are being released? How is the complement coming in to fix all this? Okay, and like I said, the interrelation with the adaptive immune system and what feedback mechanism is again the adaptive immune system giving in to ensure a comprehensive and effective response to this particular infection that might occur. Okay, so it's very, very important that we have that firm understanding of this particular concept. Okay, so we did talk about T-cell activation, and we talked about development, and we talked a bit about B-cell activation. We mentioned two types of activations. We mentioned the T-cell dependent activation, and also we talked about T-cell independent activation. So in terms of T-cell independent activation of the B-cell, we talked of a case whereby you've got either polyclonal antigens or multivariant antigens that have got a good number of epitopic expressions that are able to induce dual signals on the B cell, thus rendering that B cell active without any T cell stimulation or T cell activation. And also, this ties in at some point when we come and talk about immunological memory. We will also see how some of the B cells might be activated in the absence of a T cell when we talk about immunological memory. But more importantly, we talked about T cell dependent activation of B cell, and under T cell dependent activation, we talked of the significance of the MHC class presentation by the B cell to the T cell, the binding of the CD4 coupler mechanism receptor there, and also the co-stimulatory binding there of the CD40 and the CD40 ligand on the T cell. So that was something that we already talked about, and I hope at this point, we understand the number of signals that it takes to activate a B cell. What are those signals? What is signal 1? What is signal 2? And what is signal 3 on the B cell? And also on the other side, when we look at a T cell, we know what is signal 1, which molecules are initiating which molecules are initiating true activation of the T cell, and which molecules are further initiating additional tertiary responses of the T cell. So at this point, I believe we should have a firm understanding of those concepts. And I draw this slide for you to more provide you a basis of appreciation. And remember that in our previous lectures, we had talked about T cell activation by APCs, such as dendritic cells, such as macrophages and neutrophils. And in the previous lecture, we talked about T cell activation by the B cell. So already you can see here that the B cell itself is an antigen-presenting cell, but in terms of co-stimulatory signals, you will notice some slight changes in terms of which are the core molecules or which additional molecules are coming into play. So on my right here, we've got a B cell, and which binds an antigen binds to the B cell receptor, initiates what signal 1, and after initiation of signal 1, we've got an increased expression of the MHC class 2 molecules, and also an increased expression of CD40 receptor. Then the APC, which is the B cell here, goes and presents the peptides on the MHC class to the T cell. And then we have the CD4 receptor there confirming the binding, and we've got the CD40 receptor binding to the CD40 ligand on the activated T cell. Okay, then from there, we know that we've got a signal 2 being activated there. And then once the activated T cell is able to release some cytokines such as interleukin 2, interleukin 7, we have got that activation of the third signaling signal for the B cell. And on my right there, you'll see that we've got, again, a cell with a B cell with activation there, just like we saw. And here we see the emphasis here was on the B7 versus the CD40 ligand. So in our previous lecture, we did not talk much when we talked about B cell and T cell interaction. We did not talk much about the B7 and the CD28 binding, of which we talked about this particular binding when we talked about the other APCs such as the macrophages and the dendritic cells. So just an addition to the understanding to say that the B7 and the CD28 are also equally important in the binding of the B cell and the T cell. So it is important that as much as we have the CD40 ligand and the CD40 receptor, the B7 also and the CD28 is also very important in the completion of this particular. And so in terms of B cell activation, I don't think we have any problem with that understanding. Same as T cell activation, we shouldn't have a problem, as we have covered this in our previous lectures. Okay, looking at this slide now, I want us now, having a look at the previous slide, to have an appreciation of the key signaling differences between other APCs and the B cell, T cell, and other APC T cell. So on my right here, I have the B cell and the T cell. On my left, I have the B cell and the T cell, and on my right, I have the T cell and an APC such as the macrophage. Okay? And here, in terms of activation of the T cell, we have signal 1 being initiated by the binding of MHC class molecules to the T cell receptor. And on the signal 2 being initiated by the binding of the B7 and the CD28 receptor on the T cell. And this is the same as on the CD8. However, on the B cell, the signal transduction in terms of the second stimulatory signal is being initiated through that of the CD40 and CD40 ligand on the T cell. So here again, I repeat that here with other co-stimulatory molecules such as the B7 and CD28, signal 2 is initiated on other APCs such as the macrophages, the dendritic cells, and the neutrophils. While that of a B cell, one important molecule that we don't mention when we talk of these other APCs is the CD40 ligand, which literally brings in a second activation signal as compared to the B7 and the CD28 on other APCs. So that's quite an important difference to note that when we are talking about other APCs, such as the dendritic cells and macrophages, we have the second signal being initiated by the B7 CD28. But when we talk about B cells, the second signal activation, apart from MHC, which is number 1, the second signal is being initiated through the CD40 ligand binding on the T cell. So very, very important difference. And as we mentioned, in terms of B cell differentiation, the first signal is being initiated by MHC class. Second signal, we have the coupling of the CD40 and the CD40 ligand. And then we have got the third signal being initiated by the release of cytokines such as cytokine 2, cytokine 7 by the T cell, which further activates the B cell as the third signal signal of the B cell, which then initiates activation, increasing size of the B cell, and then it undergoes a proliferation and clonal division and further differentiation. And as we looked in the previous slide, we talked about the classification in terms of differentiation with B cell, and we mentioned that B cell differentiates after clonal division, differentiates into effector cells that these immunoglobulins such as M, and that other effector cells have different classification in which these immunoglobulins such as immunoglobulin G. And usually these type of immunoglobulins are immunoglobulins that are released upon reinfection as compared to those that are predominant IgM. So predominant IgM, these are usually released in primary immune response and predominant IgG, these are released in secondary and tertiary immune responses. And then of course, we have got the memory B cells, which we'll talk about in just a few minutes, that are also being classified. So in terms of classification, we have got the effector B cell, and we've got the memory B cell. The memory B cell, as we will learn, also differentiates into two classes of memory B cell that we will describe in just a moment. And we have here our B cell, it's B cell receptor that takes up antigen, and from then on, we have takes up an antigen. It then presents that to a T helper cell as the first signal signal. And from there, we've got the B cell, the B cell binding in terms of the CD40 and the CD40 ligand also binding, and then also the third signal, which is the release of cytokines, which further activates them. Once this B cell is activated, we've got these divisions that I just talked about. We've got production of short-lived plasma cells and memory and long-lived memory cells. And of course, when we talk of the memory cells, these memory cells, upon reactivation or re-exposure to a particular infection that the body has already encountered are able to transform into effector plasma cells, which we will see in just a moment. So this relationship is quite important to understand that once these memory B cells are generated, or under certain conditions and on re-exposure, they are able to differentiate that into effector memory cells, which then can be able to handle the particular antigen or infection that is presented. The immune system is presented. So in terms of functions of the B cell, we need to understand several functions of B cell. So of course, as we talked about, we have talked a lot about this particular binding of the B cell to the T cell, and that is the first mechanism or the first function of B cell, which is antigen presentation of the antigen peptide to the T cell. And once that is happened, we know that the second function is initiated, which is the production of antibodies, And then also we have got the production of memory cells, which is responsible for immunological memory. So here we're talking about the B cell. Okay, so at this point, we are going to dive in and talk a bit about immunological memory. Okay, so as you might know, immunological memory refers to the ability of the immune system to remember a particular antigen that the immune system might be presented. And what is important to understand that as much as we don't talk much about memory, it is a quite delicate and complex process which requires a particular combination of cytokines and a particular environment for it to be present. And so, as will be evident by the next few slides, I will show. And the memory cells usually remember specific characteristics of an antigen, and such as the shape of the antigen, particular surface molecules or proteins that are expressed on that particular antigen, so that once those antigens or that antigen reinfects the immune system, reinfects the body, the immune system will be able to remember, or the cells will be able to remember and effectively eliminate that particular pathogen that is presented. And we know that the core of immunological memory is in the B cell and the T cell memory cells, which we have previously discussed. And important to remember that during the initial phase of an immune system, the adaptive immune system does not kick in immediately. It's got the innate immune system, which responds within the first few days, and we've got the adaptive immune system that sometimes even takes weeks for it to develop an efficient response in terms of the adaptive immune system activation. And once that happens, we know that we've got the effector T cells being activated, and we've got the B cells, which then go on to produce the antibodies, which are then secreted to attack and activate other parts of the immune system. Okay, and the particular memory cells, whether the memory T cells or the memory B cell, are long-lived and they circulate throughout the body and are ready to respond at any time should there be any re-exposure to a particular antigen. And memory responses, also called secondary immune response, and tertiary immune responses, which we'll see a slide on that, all depend on the number of exposures to that particular antigen, and also the memory responses of the secondary immune response differ from the primary immune response, and they differ in terms of the size and the response to that particular antigen. And also even the location is quite important of where these particular responses happen, where the secondary responses happen, which is the memory-mediated responses, and also the primary responses. So the location and the effector mechanisms also differ between the two primary and secondary responses to an infection. And as we remember in one of our lectures, we tackled the first image there, image A, and we talked about the arrival of an infection, primary infection. We talked about the significance of this particular slide with regards to the encounter of the B cell to a naive B cell to an antigen activation of the B cell, and to the point of immune response. At what point do we have an immune response? And this particular slide will show a period of about 10 days, in which we've got a production of immunoglobulin and immunoglobulin G. And once the infection is gone, we've got a reduction in the amount of these particular globulins. And on re-exposure, we've got the memory B cells kicking in, some differentiating into effector memory cells, and we've got an increased production, heightened response. And usually this reaction has a more IgM than IgG composition of antibodies. So we see that we've got a heightened response, we've got a shorter response time in secondary responses as compared to the primary responses. And in the table below, you can see here that is classified that we have stronger secondary response as compared to primary response, but a shorter time to response in the secondary response than in the primary response. We've got a composition of more IgM than IgG in the primary response, while that at the secondary response, we've got a composition more inclined to an IgM, IgE, IgA architecture, depending on the signaling mechanism that have been initiated. And also we've got a higher affinity in the secondary responses than in the primary responses. Very, very important to remember. And on the second slide there, we re-emphasize again, looking at the specific combination that we've got the IgM composition greater than the IgG. And secondary response, we've got a stronger IgG response as compared to an IgM response on exposure. And all this is induced by the memory B cells. And this is a very important mechanism that definitely ensures the survival or the maintenance of homeostasis in the immune system. So looking at immunological memory at work, we've got a description there again, which is showing the effect of the primary response, secondary response, and the tertiary response. In blue there, we've got the IgM primary response there, and then we can see how the composition of the IgM fluctuates over the course of the primary and secondary infection. And we also have the response of the IgG in a primary response, in a secondary response, and in a tertiary response. So we see that at each point, the response is being heightened, and the time to response at each point is being reduced, so that we always have a stronger, faster, and more efficient immune response to re-expose the antigens at any time. And this is the concentration there. And also when we look at the affinity of the IgGs and IgM, we see that on re-exposure, we always have a stronger affinity of either the IgG and the IgM. And of course, a higher concentration of IgG than IgM exposure. And this basically shows the concept of smallpox. After the smallpox vaccination, antibodies level show a significant decline. T cell memory usually have a lifespan of about 8 to 15 years for smallpox, and usually for some people, even develop lifelong memory for this particular type of infections. And to re-emphasize again in terms of the affinity and the concentration and the composition of the immunoglobulins, we've got here. Here we've got the frequency of antigen-specific B cells. We see that in an immunized donor versus an unimmunized versus immunized, we see that we've got a higher response in the immunized as compared to the unimmunized. We also have got a different architectural composition of the immunoglobulin, more inclined to the IgM in the unimmunized individual, and more inclined to an IgG, IgA in immunized individual. And affinity, as I mentioned, we've got high affinity in the immunized and low affinity in the unimmunized. And this basically is the same as when we talk about infection versus infection versus reinfection. Okay. And of course, here we want to talk about somatic hypermutation. We know that the hypermutation is high in the immunized or on reinfection because due to the increase in the class switching of the immunoglobulins in order to produce the IgG, IgA, and IgE in response to that particular type of infection. While here, predominantly, we just have the IgM immunoglobulin. So the class switching is very, very low in the immunized or in the primary response. And again, we try to re-emphasize the concept of antibody specificity, antibody affinity. And here we show exposure at 2 years, we show exposure at 5 years, and we also show the exposure at 20 years. And you can see here we have an individual infected with influenza virus making antibodies against all the epitopes that are present in the virus. So, but that virus infects an individual, and those are the epitopes, or those are the viral epitopes that are present, and the individual makes antibodies against those at the primary response stage. 5 years later, we have this individual. They are re-exposed with a variant influenza virus, which has got some slight mutations or differences as compared to the first one, but with some similarities in terms of some epitopes. And you can see here that when we look at this epitopic presentation, we see that A and C are also present, were also present in the first variant of influenza that was there. So in that response, the immune system is able to respond, is able to identify all those epitopes that were identified in the primary response. It's able to identify them and heighten the response. And here we've got a B and a C having a high response in the secondary response after 5 years. And here again, after 20 years, this individual again is exposed to another variant, a new variant of the same influenza virus. And this variant again also has slightly different epitopes on its surface with similar epitopes, with a few similar epitopes to the parent variant. And again, we see here that the individual is only able to produce a high response to those epitopes that were present in the parental variant. And we can see here our B, our A, and our D having a heightened response as they were also present in the parental variant. So very, very important to remember. Here we are trying to basically show the concept of specificity of how the immune system is responding to a particular antigen on re-exposure, but a primary, secondary, and tertiary exposure. And this individual, once exposed to a variant virus, is only able to produce immunoglobulins against the immunoglobulins that were present in the variant or what we call the parent variant. And those are the immunoglobulins they were able to produce. And again, on tertiary exposure, we see that they were only able to produce immunoglobulins against the epitopes, viral epitopes of that were similarly present in the parental variant. So very, very important to remember that the immune system always remembers, and apart from remembering, it's always able to launch a stronger, as you can see with A, that A response to the A epitope there, you can see the level there. Upon secondary exposure, we've got an increased heightened exposure, and on tertiary exposure, the amount of antibodies being released increases further, similar to what we saw in the previous slide. So we've got that increase, and also for C, we've got that increase in the level of exposure, which leads to the increased level of antibodies being produced by the B cells. And like we covered when we talked about the B cell activation and differentiation and the T cell. So here in terms of the T cell, we have got a naive T cell there coming in contact with an antigen presenting cell. Let's assume, for example, that's maybe a dendritic cell, which presents the antigen to the T cell. So once the T cell encounters an antigen, it undergoes activation, and from activation, we've got most of the T cells becoming effector cells, and some of them differentiating into memory T cells. And usually the memory T cells have got a unique classification or unique increased expression of interleukin-7 receptor. And when we talk of the effector cells, many effector cells are short-lived and will die by apoptosis if they are not engaged within the immune system within a short time. However, with the memory cells, we've got cytokines such as interleukin-7 and 15, which are responsible for maintenance of the survival of the memory cells within the immune system. And of course, with a particular expression of the interleukin-7 receptor, which maintains the particular dominance of these particular memory cells. And these memory cells, for their maintenance, they will need to come into contact with the MHC self molecules in order to maintain their population within the immune system. On my right there, basically, we describe the differentiation of the memory cells, of which, of course, we've got the naive cell binding to a dendritic cell, and then dividing, of course, into an effector T cell, which then differentiates to a memory T cell, as we have expressed there with the classical interleukin-7 receptor alpha on the surface and the CD45 RO receptor on the surface. And as I mentioned, the memory B cell, the memory T cell, then differentiates into two classes. We've got the central memory cells, which usually express the chemokine receptor 7, which usually remain in the lymphoid tissue, and then it also differentiates into an effector memory T cell. So these effector memory Ts, the central memory T cells, once reinfection occurs, are able also to redifferentiate to provide support to the effector memory cells. As I mentioned, when we talk of classification of memory cells, we've got the effector memory cells, and we've got what we call the central memory T cells that are within the lymphoid organs. And these other effector cells are within the tissues and are migrating in order to detect any possible antigens within the lymphoid organs as well as in the periphery. So this is an important classification that we'll need to remember in terms of differentiation of the memory cells into the central memory cells with the classical expression of CCR7 within the lymphoid tissues, and also the effector cells, which are within the periphery and other tissues that have got a classical expression of CCR3, CD45, and also CCR5 receptors on their surface. Okay, so that leads us now to talk about what are the benefits of immunological memory. And as I mentioned, faster, stronger, specific. Those are the main 3 points that we need to carry on today. Faster, stronger, longer. Okay, so we've got the faster response. Immunological memory allows our immune system to respond more quickly to familiar antigens. That's ideally on exposure. This rapid response can help prevent the infection from spreading and also reduces the severity of the particular infection of the illness. When we talk of long-lasting protection, this memory can live for a long time. And like I said, some of them can even live or can be present throughout the lifetime of an individual, providing protection against pathogens that might reinfect the particular individual. Okay, and also they are very specific to particular targets or specific antigens or antigenic epitopes that might be encountered. And this specificity allows our immune system to distinguish between harmful and harmless antigens, and also to minimize the concept of any chances of unnecessary immune responses. Okay, so like I said, it's faster, longer, and specific. Those are 3 main important benefits that the concept of immunological memory provides for us. And as we talked about in a slide before this, the most important comes in when we talk of immunological memory is the concept of vaccinations. When we receive a vaccination, our immune response is exposed to a weakened or inactivated form of a particular pathogen. And this exposure triggers production of the memory cells, both the T cells and the B cells. And once those T cells and B cells are active, we've got the memory cells coming in. And should we be exposed to an actual pathogen that carries similar epitopes on its surface, we are going to have a faster response, a specific response, and also we're going to have a longer response that will protect us for a longer period of time. And like I said, the other most important concept is the concept of reinfection or repeated infection, which protects us against those. Okay, so to just conclude the summary of immunological memory, we need to know that immunological memory is a remarkable defense system or mechanism of the immune system. It allows us to remember and efficiently fight off harmful organisms that may be encountered, and the presence of immunological memory provides a faster, specific, and longer response or protection against any type of diseases that we might encounter. And this particular understanding or appreciation for immunological memory has helped scientists and healthcare professionals to develop vaccines, treatments, and strategies that ensure that our immune system is able to respond in order to protect our own well-being. So that would be the small conclusion on immunological memory. Any questions you can ask at this point. Anything on immunological memory that needs clarification? Okay, let's proceed. Okay. So, like I said, we've already talked about the binding or the concept of antigen presentation of the B cell. We have now talked about immunological memory, and we hope we have a good understanding. We've also added T cell memory to that concept. And now we are also now going to talk about antibody production. Okay, we're going to talk about antibody production and add to what we previously covered in the introductory classes for this program. Okay, so as you might remember, we have 5 classes of immunoglobulins, which is the M, A, G, D, and E. And the focus of this particular lecture on antibody molecules is basically to appreciate or understand the unique structure in terms of the constant region and how it interrelates to its function, and also appreciate the different structural variants of these individual molecules and how they relate to their function. This becomes important as we handle specific infections within the immune system. We will see how the significance of immunoglobulin M, for example, the significance of immunoglobulin M, what is the significance of the structure of immunoglobulin G and E and the others. And this all ties in when we come to the description and trying to image how the immune system responds to particular types of infection. Okay, so as you previously covered, the first immunoglobulin we're going to talk about will be the immunoglobulin M antibodies, IgM antibodies. And ideally, as we saw, this is the first immunoglobulin that is produced from B cell development, being added to the immature B cell before it leaves the bone marrow as a transient B cell. And also we talked about the mature, a naive mature B cell having a classification of the immunoglobulin receptor. And then of course, as it becomes a mature B cell having the immunoglobulin D. Okay, so production of the IgM, as we saw in the previous slides, on primary immune response, this happens usually during a primary immune response where we get the heightened expression of the M. And immunoglobulin M are good at activating the complement, and some of immunologists have ended up coining this as a fixing complement antibody, due to its unique pentameric structure. As you can see, its unique structure, it literally has got more of 5 immunoglobulin anchored by the J chain and bounded by the disulfide bonds. And usually this particular immunoglobulin has a half-life of about 1 day. Okay. And there we have it there. We have got the basic units there of immunoglobulin with a light chain and a heavy chain. We've got the J chain there and the disulfide bonds holding the IgM immunoglobulin. So a key question that we ask is, how does the IgM antibody work in antibody-dependent activation? So as you might remember, when we covered the complement system within the blood and tissue, some of the complement factors get together to form a big complex called C1. So despite its size, this complex of protein cannot activate the complement system because it is bound to an inhibitor. And due to the unique structure of the IgM molecules, this C1 molecule is able to bind to the constant regions of the IgM molecule. And once bind to the constant region of the IgM molecule, it allows for the inhibitor to drop off, thus leading to the activation of the C1 complex, which then leads to the activation of C3 convertase, okay, which then activates the complement system, or the classical pathway of the complement system, which we already covered. So the unique structure of the IgM molecule itself with its unique pentameric structure, consisting of a number of antigen binding sites and also a number of constant regions. Here we can see we have got about 5 constant regions where we are able to bind complement, and once this complement are able to bind there, they move closer to each together, dropping off the inhibitor there, which is important in the activation of C3 complex. And like I said, this activates the complement system in terms of the classical system, which we often refer to as the antibody-dependent complement activation. Okay, so this is basically what I described here, where the IgM molecule are able to bind antigens via the antigen binding site, and also to the C1 complex, which bind to the FC region, constant region of the antibody. And the constant regions, we've got 5 constant regions, and then 10 antigen binding regions on the IgM molecule. So then, because of that unique structure of the IgM molecule having a lot of binding sites on its surface for antigens as well as for other components of the immune system, such as the complement, it's very, very significant in early infection, as it might be able to activate complement and also bind to a number of antigens or pathogens within the context of the primary immune response. And this is where we always mention to say the B cell is the main linker between the adaptive and the innate immune system, because at the end of the day, we've got a complement system binding to immunoglobulins, which are produced by the B cells, which further enhances activation of the innate immune system as well as of the adaptive immune system. So the IgM molecule itself, due to its structure, is able to neutralize, bring in a state of neutralization due to the number of antigenic binding sites that it has, is able to bind a number of antigens, therefore bringing in a state of neutralization so that those antigens are not able to cause infection. And no wonder the IgM is always the first antibody to be released at the site of infection, because of its unique structure that we have just described. So the B cell, as we know, produces a lot of antibodies, which will eventually coat bacteria or foreign entities that might infect the body. So apart from the binding to the complement itself, we've got the immunoglobulins, which are binding to particular surfaces of bacteria. And as they do that, together with the complement, which is also bringing together a number of complement molecules together, it's able to extend the range of the activity of the immune system, as it is able to ensure a state of neutralization within that particular infection. And it's very, very important that we understand that immunoglobulin M is one of the significant molecules or antibody that activates the complement. And that's why when we talk about, from a straight perspective, when we talk about the name complement, it literally comes from a state where immunologists, when they were doing experiments, they noticed that most of the antibodies were more effective when they were in the presence of a particular protein. And hence these proteins were therefore classified as complement, because they complemented the activity of the antibodies, and hence the classification of the complement system from those standpoint. When you talk of the IgG molecule, of course, the G stands for gamma. We should know that the IgG is the most abundant immunoglobulin in the body. Okay, and we have got different structural isoforms of the immunoglobulin G, and as described there, we have four classic presentations of the IgG molecule. What are IgG1, 2, 3, and 4, all which are structured different from each other. And you see that this constant region, its difference also conveys some form of functionality, determines what type of antigen these particular IgGs are able to respond to, as we will see in the next slide. So most importantly, IgG3 fixes complement better than any known IgG molecule. And ideally, this is due to its unique structure. As you can see, that's got a long constant region. So which means that the complement factors are able to bind to the constant region, which then initiates activation of the complement system, apart, of course, from the IgM that we have previously talked about. Then also, we've got IG1 subclass antibody, and this has been classified as an important binding or linker in the opsonization of invading pathogens. Okay, this is because macrophages and neutrophils have got specific receptors that are able to bind with high affinity to the FC region of the immunoglobulin, of the IgG immunoglobulin. Okay, so just to repeat that, the immunoglobulin IgG1 has got specific receptors that are where the macrophages and the neutrophils are able to bind. And these binds, remember that these are antigen-presenting cells, these are phagocytic cells. And once the IgG, it binds to particular invading pathogen in a way of opsonization or coating, these macrophages are able to identify the IgG1 and therefore initiate a process of phagocytosis of that particular pathogen. So we've got IG1 very good at optimization, we've got IgG3 very good at fixing complement within the context of the net system. Okay, just to take it further, also we've got the natural killer cells also have got their receptors that are able to bind to the IgG. So apart from the ability to fix complement, we've got the ability of the IgG3 being able to become a linker or initiate NK natural killer cells killing mechanisms. Okay, so the IgG3 ideally would become a bridge between the NK cell and the target cell or the viral infected cell via its variable region on the infected cell or on the antigen, and also it has unique receptors within its constant region, which binds the FC receptor on the NK cell. And this brings in the concept of antibody-dependent cellular cytotoxicity, ADCC, which is initiated via the NK cells. So the NK cell does the killing, but the antibody identifies the target. Okay, so antibody via its FAB region is able to bind to the antigen, which is on the surface of the target cell of the infected cell. And once that occurs, the NK cell is able to bind to the constant region of the antibody, and therefore is informed or is instructed to induce its activity in terms of identification of the target cell. And this is in a concept that we refer to as antibody mediated or dependent cellular cytotoxicity. So very, very important concept to understand further. Yeah, so and just to re-emphasize again, so therefore immunoglobulin Gs also have got a fixing, a good at fixing complement and also neutralizing viruses. And we can see here in terms of antibody-dependent cellular cytotoxicity, we've got an infected cell that has got a lot of immunoglobulins that have bound to the antigenic surfaces on the cell. We've got the NK cell with its FC receptor binding to these particular antibodies. And in this case, we'll be talking about IgG3. And then from there, we've got an induction of apoptosis of cell lysis as initiated by the NK cells. And on the other hand, when we talked about opsonization, again, we have got the immunoglobulins binding to the surface of the particular pathogen and then marking it for destruction. As you can see, we've got the phagocytic cells here, the macrophages and the neutrophils, which then bind to the FC region of this particular immunoglobulins. And once they bind to the FC region of these particular immunoglobulins, they're able to initiate a process of phagocytosis. And also, we've got the concept of complement activation from that end. Okay, having talked about the IgGs, it is very important to know that due to its unique structure, the IgG antibodies are able to pass through the maternal blood through the BBB, the blood-brain barrier, and also into the blood of the fetus by way of the fetus. Okay, very, very important to remember. The IgG molecules are able to pass through the placenta into the blood of the fetus and are very, very important in the first weeks of life of the infant. Okay, this provides the fetus with a supply of Ig antibodies that will protect it for several weeks to months as the infant develops its own immune system. Okay, so a good question for the class, and be free to answer. So we're going to talk about IgA molecules, and there's a question here. Which is the most abundant antibody in the human body? I think we talked about something in the previous slide. Is anyone able to tell us which is the most abundant antibody in the human body? Anybody able to tackle that question? Which is the most abundant antibody in the human body? Go around the poll. Which is the most abundant antibody in the human body?
Sir, I feel it's IgG.
Okay, awesome. Anybody else?
Yeah, I think it's IgG. I remember you mentioned it.
Okay, that's Douglas. Anyone else? Can we have a third one to confirm?
IgG.
Awesome, awesome. Okay, so this is a very, very good question that we always bring in one way or the other. In the previous slide, I mentioned that the most abundant antibody in blood was IgG. Okay, let's go further. No, it's not IgG. IgG is not the most abundant immunoglobulin in the body. I mentioned that it is the most abundant immunoglobulin in blood. And no, it's not IgM. Okay? So I want us to be very, very distinct as we describe the most abundant antibody in blood is IgG. In blood is IgG. However, let's go further. The most abundant antibody in the body is IgA. I hope I'm not confusing you there. There are two things we're describing. We're describing abundance in blood and abundance in the body as a whole. So when we talk of blood, we're talking of IgG. When we talk of the entire body, we're talking of IgA. And how's that? Okay, so it sounds like a trick question. And usually, if we don't bring this to light, most people, even fellow scientists, will try to get that one wrong. Okay. And remember, I have mentioned that IgG was the most abundant in the blood, which is very, very true. Okay. It's abundant in the blood in high concentration. But when we talk of the entire body, it is actually IgA. Okay. So it turns out that our human, that we as humans synthesize more immunoglobulin A antibodies than any other subclass combined. Mr. Clayton Peary, you have a question?
Yes, sir. Like, yeah, what is the question? Just explain, like, what is the abundance in immunoglobulin? Can you answer A also?
Repeat your
specified maybe blood.
Repeat your question.
Like, what if the question is just general? Like, what is the most abundant immunoglobulin? Not mentioning in blood or in the body.
So if the question was to be stated like that, I would still go for IgA.
Okay, thank you.
You understand?
Yes, sir.
Yes, because we're talking about the body as a whole. So that would definitely be IgA, and I'll explain why in just a minute. But if we talk of blood itself, or somebody would say blood in serum, in a plasma, we will talk of IgG. But we talk of the body as a whole, or if we left it blank to say which one is the most abundant antibody, definitely go for IgA.
Thank you.
Okay. So the last bullet there is that the human body synthesizes more IgA antibodies than any other immune classes combined. So if you combine all the subclasses of the immunoglobulins in terms of total in the body, IgA still has the most abundant concentration in the body. Okay, and the reason that is there is because the IgA antibody, okay, provides protection for all mucosal surfaces in the body. And remember that in terms of mucosal surfaces, this ranges to about 400 square kilometers. And the immunoglobulin A is found in all these mucosal surfaces that are lining the body. Okay. So when we talk of quantity, if we look at the mucosal surfaces that are in the body and then calculated them in terms of area, we come close to about 400 square kilometers of mucosal lining. And all these mucous membranes and all those all have got the presence of IgA. And under the surface, there is a specific type of B cell which is producing those type of immunoglobulins in order to protect our body. Okay, let's go further. So there is a description of the mucosal tissues of the body. So when you talk of mucosal tissues of the body, I started from my right. We are going to talk about a number of systems, literally all systems, from the respiratory system to the oropharyngeal system. Okay, we'll talk about the reproductive system, the renal system. All these areas have got mucosal lining. The eyes, all those areas have got mucosal lining. And the most important immunoglobulin that is lining all these surfaces is immunoglobulin A. Let's talk about the intestines, the stomach, the esophagus, okay? The trachea, the lungs, the sinuses, the oral cavity, they also, name it. And when all these mucosal surfaces are calculated in terms of area, we rank up about 400 square kilometers. And these immunoglobulins, specific immunoglobulin, and this particular case, immunoglobulin A, are present in all these surfaces in high concentration. So when we combine all the immunoglobulin subclasses on one hand and the immunoglobulin A on one hand, and did the concentration, we find that the concentration of immunoglobulin A is high as compared to all the other subclasses combined in the body. But when we talk of in the serum, in blood, in plasma, okay, already we know the difference between plasma and serum. So some people would say, okay, what is the concentration of, they are most abundant in blood? You say, oh, it's IgG. Then they'll say, okay, so tell us what is the concentration in serum, which is the most abundant antibody in serum. It is still IgG because these are components of the blood depending on the condition in which that blood is handled. If it is allowed to hemolyze, what do you have? You have serum. If it is not allowed to hemolyze, what do you have upon separation? You have plasma. So let's always remember most abundant antibody in the blood is IgG, and the most abundant antibody in the body is IgA. And I've given the reason why. And this is because the IgA immunoglobulin lines all the mucosal surfaces of the body, which ranges to about 400 square kilometers of mucosal surface. And therefore, these IgA immunoglobulins are even referred to as defenders of the mucosa surfaces. Okay? Very, very important distinction to make. Okay. And the question that will come based on that structure comes back to the issue why we're talking about immunoglobulins. The issue here is the structure. Okay, as you can see in terms of structure, you literally have two primary immunoglobulins back to back held together by a clip. Okay. And in terms of classification of the population of B cells, about 80% of the B cells that are located underneath the mucosal surfaces produce immunoglobulin A. Okay, so imagine all the surface, all the B cells that are under these particular surfaces, 80% produce immunoglobulin. And that makes immunoglobulin A one of the most abundant immunoglobulin. And that comes back to its unique structure that it has. And like I've said, you will find that you have got the basic, what you call the basic unit of an immunoglobulin 1 and the other basic unit held together by the J chain or the clip, as it's also referred to. So one reason why the immunoglobulin A is so good at defending against antigens is that it's able to penetrate mucosal surfaces, and that is due to its unique structure or connection here. Okay, this structure allows it to easily pass through mucosal surfaces without any challenge as compared to any other immunoglobulin type. Okay, and as you will see, also the constant region of the immunoglobulin A itself is also significant in this particular mechanism of action of the immunoglobulin. And there we have a schematic description of a plasma secreting, an antibody secreting cell in the submucosa, releasing an IgA dimeric structured immunoglobulin. And we can see there that the immunoglobulin is able to move at ease through the mucosa into the lumen of the gut without any challenge. And it's the only antibody that has got this unique ability to do that. And that highly relates to its particular function. Okay, so the particular mechanism here of the J chain or the clip mechanism is more of a passport, so to say, for it to easily maneuver through the mucosa lining after it's produced from the particular specialized business. Okay, so it's more of a special permit or passport that allows it to easily move through. And because of its unique structure, it's able to resist most acids and enzymes that are found in the digestive tract. That's also one significance of that J chain dimeric structure arrangement. And due to its flexibility as well and structure, it's also it's primarily able to bind onto the surface of any invading pathogens and prevent those pathogens from infecting the lumen or the mucosa surface that is being protected in whichever part of the body. So once the secreted immunoglobulin, once the immunoglobulin is secreted, moves through the epithelium, it's able to bind to the invading pathogens, and those pathogens will not be able to infect the mucosa surface within that particular region. Okay, and as I mentioned, this is due to its unique dimeric structure, where it has got the clip of the J junction here, which is very, very flexible, and also it has got specific receptor component within this particular FC constant region that is able to easily penetrate, easily move through the epithelium into the area that it is needed most. And because of its structure, having a dimeric structure, it's able to bind different or a number of pathogens. Having a dimeric structure allows it to literally bind from its antigen binding region to bind at least 4 key molecules that are able to bind to those particular region. And then also with its unique structure in terms of the FC region there, it is very, very effective at causing the rejection of bacteria. Since this bacteria now cannot enter or infect, it will end up being flushed out together with the mucus within the lumen in that particular surface. And just a reiteration there, you will find that in fact, rejected bacteria make up about 30% of normal fecal matter. So imagine fecal matter being released, and 30% of that is actually rejected bacteria, and that bacteria cannot cause infection within the within the body because it is bound to immunoglobulin A. Okay, and as I mentioned, because of that particular system or mechanism, it is usually referred to as because of a guardian, or others would say these are guardians of the mucosa surface. Okay, and also quick to note is that immunoglobulin A is secreted in breast milk, so very, very important, and probably due to its structure and function and its location, it's quite significant that it is produced in breast milk. And the reason ideally is that the IgA molecules coat the baby's intestinal mucosa and prevent or provide protection against pathogens that the baby might ingest. Okay, and this is critical, as many of the microbes within the environment that the baby encounters are taken through the mouth. So therefore, the milk that the baby drinks from the mother will contain a high concentration of immunoglobulin A, which coats the epithelial lining or mucosal lining to ensure that whatever pathogens that might be taken in by hand, mouth, are not able to cause infection. And one important concept that we will need to remember is that as much as the immunoglobulin A are effective in fighting off mucosal antigens or invaders, they are quite useless in terms of complement fixation. And this is a very, very good point. Can anyone tell us why the immunoglobulin A is useless in binding complements? So complement C1 factors will not bind to the constant region of immunoglobulin A. Would anyone have an idea why immunoglobulin A does not bind or does not play a significant role in complement fixation or activation of the complement system? Okay, very, very good question. And you need to remember that question. Okay, so we'll go back to the concept of immunoglobulins. And like we said, when we talked about immunoglobulins in our introductory classes, we know that the constant region of the antibody determines its class and its function. Okay, so it's very, very, the constant region on immunoglobulin is very, very important in determining the class of the antibody and also its function. And the lack of the complement fixing activity or complement binding activity of the immunoglobulin is actually a protective mechanism, or let me just say it's a good thing. Okay, and why is it a good thing? Okay, and the reason is quite simple. Okay, and I have stated there, if immunoglobulin A would initiate complement reaction or activation, our mucosal surface will be in a constant state of inflammation in response to all those pathogenic and non-pathogenic entities that are constantly going through the mucosal surfaces. And because of that, we will be in a state, the mucosal surfaces will be in a state of constant inflammation. Imagine your intestine will always be inflamed if immunoglobulin A was able to activate the complements. And of course, if we are in a state of inflammation all the time, I don't think that's one a good thing, and it's literally pathological in itself, being in a constant state of inflammation. Okay, so that's why the immunoglobulin A functions more of a passive antibody that basically is responsible for blocking binding to pathogenic entities that are coming through the mucosal lining, wanting to invade the mucosal lining. And as it binds, it makes sure that it initiates a process where those particular pathogens are not able to infect the particular linings and are able to be flushed out of the particular region they are in, whether it's the eyes, the intestines, or whatever region where there is a mucosal surface. Any questions on that? Why immunoglobulin A does not bind or activate complement? It's a very, very important question to remember. Okay, immunoglobulin A does not bind complement because, one, if it did, the body or the mucosal surfaces will be in a constant state of inflammation, which in itself is pathological. The basic function of the immunoglobulin A, which is encoded in its FC region, is basically to bind potential pathogens and ensure that those potential pathogens are flushed out of the system wherever that mucosal system or mucosal lining is. I hope that's quite clear. So always remember that patient, very, very important patient. Okay. Then when we talk about immunoglobulin E, again, with regards to the basic structure of an immunoglobulin, you can see I have compared the immunoglobulin E to the immunoglobulin G in terms of structure. And I'll need you to note a very important structure in the constant region of the immunoglobulin E that also will be significant in the role of immunoglobulin E. Okay, so immunoglobulin E has a molecular weight of about 200,000 DA. It's a very rare immunoglobulin. The serum concentration is about 10 to 400 nanograms per ml. And in terms of total body concentration, it is literally about 0.002% in terms of concentration within the body. Okay. The immunoglobulin A has a high affinity for allergens and IgE receptors. Okay, IgE receptors such as the Fc epsilon receptor that is primarily on the eosinophils and the basophils. Okay, and I'll come back to the reason why we have an IgE molecule that has got a variant or different structural combination of the Fc region to that of the basic unit, which is the IG molecule. And just from here, you will see that it has got three regions in its, it has an additional constant region as compared to the basic IG molecule. And we can see here, in terms of the heavy chain, it has got three heavy chain units in its constant region as compared to the standard immunoglobulin class, which has got two heavy chain units. Okay, and I'll repeat this. The immunoglobulin E has got a high affinity for allergens and immunoglobulin E receptors for allergens and IgE receptors. Okay, so here in I go further to state that the heavy chain contains an extra domain, which is the C4 domain. Okay, where it attaches with high affinity Fc epsilon receptors, which are found on basophils and eosinophils. Okay, and most of these basophils and eosinophils are found in saliva and nasal secretions. Okay, let's go further. And also note that immunoglobulin E and D, in terms of the composition, we've already talked of the percentage of 0.002% of the immunoglobulin E. Also, similarly, immunoglobulin D also has a very, very low, low and similar concentration within the body. Okay, we're still talking about immunoglobulin E, and here we basically show the T cell and B cell interaction leading to the production of an IgE antibody. Okay, so there, as we have described previously, we have the T helper cell and we have the B cell. And once the B cell, which is an antigen-presenting cell, has got an allergen, okay, that has bound to the receptor, we've got an increase in the expression of the Ig of the MHC class molecules, and therefore an expression or an increased expression of the CD40 receptor. And once the allergen is presented to the T cell receptor, we've got activation of signal transduction within the T cell, which then initiates the receptor binding of the CD40 ligand to the CD40 receptor. And once that second signal comes in, there's activation of the third signaling molecule on the B cell, which will ensure a DNA recombination. Okay, and then we have all the other processes coming in to ensure that we have complete B cell activation. And also the third signaling molecule, where we have got the release of interleukin-3, interleukin-4, interleukin-13 being released to further activate the B cell for further signal transduction, which will lead to the production of an IgE molecule. This combination that you see, this interaction here, will become very, very important as we look at what happens when the immune system is presented with, for example, a parasite, okay? And we know from our hematology that when we are presented with parasitic organisms, what type of cells are activated and what type of immunoglobulin are activated in response to parasitic infection. Okay. So therefore, we talk from that context, we then talk about immunoglobulin E-mediated immunity and inflammatory responses or reactions. Okay, so this usually occurs primarily in response to allergic reactions and usually to parasitic infections. Okay, such as protozoa and other parasites that will initiate immunoglobulin E inflammatory reactions. And in terms of immunoregulatory functions, we have got the regulation of immunoglobulin E receptors, promotion of mast cell survival, suppression of innate immune responses, and enhancement of T cell responses to allergen. All these are part of immunoregulatory functions of the immunoglobulin E. And more importantly, when we talk of the mast cells, when we talk about immunoglobulin antibody-induced mast cell activation, we already know that stored within the mast cells, we've got a vast amount of a lot of granules that have got a lot of chemicals, and one of those important chemicals that we have heard of is histamine. Okay, and when a mast cell encounters a parasite, and sometimes in response also to allergic reactions, these contents from a mast cell are released and then lead to a destruction of that particular parasite. And due to that destruction, which is non-specific, and remember that we're talking about a mast cell that is from the innate lineage of the immune system. The action of the mast cell is non-specific. And as we talked about, the adaptive immune system is very specific. So the mast cell, once it encounters these parasites or in response to an allergic reaction, would release this cocktail of chemicals that are within its granules. And these will lead to destruction of that particular pathogen. And also, apart from that, it will lead to what we call local destruction of the tissue in which those particular contents have been released. And in doing so, it causes a harmful effect on the body and usually causes an anaphylactic shock. if it is very excessive, if the response to that particular allergy or infection is very strong, you end up having an anaphylactic shock. And there we are showing a structure of a mast cell with its unique IgE receptor. Okay, and as we mentioned, that the immunoglobulin E has got receptors in its Fc region that are specific for the immunoglobulin receptor that is found on the mast cells. Okay, and here we're showing a region, a concept in which the immunoglobulin is armed. This is in response to a secondary immune response. So let's go a bit deeper. How does this immunoglobulin E mediated response work? Okay, so in terms of let's say an allergic reaction, for example, we have a B cell there with its B cell receptor has been exposed to a particular antigen, and in this particular case, we have a pollen antigen that has attached the B cell receptor, and as per usual, this B cell has undergone activation, and after activation, this B cell has produced the IgE immunoglobulins. Okay, so this is as a primary immune response to this particular pollen antigen. So the B cell has produced these immunoglobulins, and these immunoglobulins have got a specific affinity for mast cells. And remember that once these immunoglobulins are released, these immunoglobulins have got an affinity or higher affinity towards the initial antigen that initiated the activation of the B cell. So the specificity of these immunoglobulin is towards the initial parental antigen that initiated activation of the B cells. So once the immunoglobulin E, the IgEs are released, they form and bind onto the surface of the mast cells, which has a high affinity for them. And once they bind the mast cell, once the mast cell comes into contact with that same or highly similar antigen, we have got activation of the mast cells. And once the mast cell is activated, it then releases all its chemical components or chemical transducers within its contents, and then releases them into the particular microenvironment in which it has encountered that particular antigen. And this is what then now causes this array of symptoms, and sometimes what we refer as an allergic response. So that is basically how you have an allergic response. You've got pollen, first encounter with the B cell, activates the B cell, and upon activating the B cell, we've got immunoglobulin E being released, which then binds with specificity to the mast cell. The mast cell then, once it comes into contact, and here at this point, once the immunoglobulin E binds to the mast cell, we usually say that the mast cell is armed already, or is, let's say armed already. And once it is activated by this immunoglobulin, it releases all these chemical contents, and histamine being one of the most significant immunoglobulins on its surface. So once it releases these, we have now all these symptoms that we have, and usually we have got these symptoms that are characteristic of an allergic response. And on my left here again, I basically just try to show a more descriptive binding of an allergen to the B cell, binding of an allergen to the immunoglobulin E receptor on the mast cell, which leads to the degranulation of the mast cell, and therefore leading to an allergic response or allergic reaction. So very, very important to remember how the immunoglobulin E antibody induces an allergic response to an antigen such as a pollen antigen. Okay, and here basically we show the structure of a the structure of a mast cell. And as you can see on the top there, we've got the immunoglobulin E antibody, which have bound to an antigen. And once those the antigen binds to the immunoglobulin E on the surface of the mast cell, we've got an activation of a signal transduction process that moves from the conversion of ATP into all these other different stages there, leading to the release of all these chemicals such as heparin, and the most common usually that we talk about is histamine. And on my left here, we've got a mast cell that is armed, and on my right here, we have a mast cell that is undergoing degranulation, short an activated mast cell, and on my left, we have got an armed mast cell. And this is the process in which an antigen goes through to initiate an allergic response. Okay, so that's immunoglobulin E. In relation to the other immunoglobulins, already we can see in terms of molecular weight, okay, in terms of half-life in days, and all the other activities. So in terms of activity, it mediates hypersensitivity. Does it induce antibody-dependent cell cytotoxicity? Yes, it does. Does it fix complement? No. Does it ensure antigen neutralization? No. Okay. Okay, that is on immunoglobulin E. And when we come to immunoglobulin D, also we can see similar, almost similar half-life to that of immunoglobulin E, with a slightly similar size in terms of kilodaltons when we talk of molecular weight. And the IgD maintains the double domain structure of the classical immunoglobulin structure. Okay. Then we remember that when we talked of mature B cells, we found that the immunoglobulin D was expressed on the surface of the mature B cells. Okay, so from this slide, what is important to note, first of all, is a co-expression of the immunoglobulin IgM and IgD on the surface of mature B cells, and also the specific presence in that it makes about 1% of all proteins in the plasma membranes of B cells. Okay, that is slightly important to remember. With regard to function, the antibody receptor, the antigen receptor on the B cell is involved in regulating B cell function when it encounters antigens. And of course, the most important is when IgM and IgD expressed on the B cell interact with an antigen for which they have specificity, the antigen is internalized. Remember that the B cell is an antigen-presenting cell. It's a cell that is among the APCs. It internalizes antigens and presents them to the T cell. Therefore, the CD4 receptor is also significant in this process of internalization that ensures the proliferation and differentiation of the B cells into the plasma cells. Okay, then the secreted immunoglobulin also exists and plays an elusive function in blood because of secretions, and also on the surface of innate immune cells such as the basophils. Okay, as I've mentioned, it binds to the basophils and mast cells and activates these cells to produce antimicrobial peptides or factors that are functional in the respiratory immune defenses in humans. And is also a significant factor in allergic reactions. So looking at that slide there, I hope it's clear from your end. I'll see if I can get a better image. But basically here we're trying to describe what is happening with regard to this particular immunoglobulin that we're talking about. So this is basically a whole system here in which we've got the innate side of it versus the adaptive side of the immune system. Okay, so one thing we need to note is that first of all, if we have where do we start it? Let's start it from up here. Okay, so first of all, we can see here we have the B cell here. This is a mature B cell. We have the dendritic cell. We have the monocyte there. We have the T helper cell, and we have also these B cells. Okay, this is a plasma cell here, and this is a basophil. Okay, so once we have activation of this particular B cell here, we've got this transformation into an IgD secreting plasma cell. These IgD molecules are essential also in mucosal defense, and also they are essential in activating or calling the basophils to produce further chemical signals and also to come closer to the site of infection. Okay, so migration of the basophils initiated by the activity of immunoglobulin D, these then move towards the site of infection under the influence of interleukin-3, and the basophil itself also then undergoes a number of processes. Once it is active, it reduces a number of antimicrobial defenses, such as interleukin-37 defensins, which are essential with the destruction of pathogens within the area of infection. It also produces pro-inflammatory factors. And also here we've got other chemotactic factors, which draws a number of immune cells to the site of infection. And also it has got other antibody-inducing factors which ensure the activation of the B cells, so that we've got more plasma cells being produced. Here we've got the T cell and the B cell activation happening there. And apart from that, we also have the dendritic cell, which is presenting through the TCR, presenting to the T cell. We also have got the activation of the B cell here by the T cell. And once the B cell is activated, we have got this process that happens with regard to the immunoglobulin D. Okay, so we have got pathogens. I'll go through again. We've got pathogens that are detected or antigens that are being detected. The dendritic cell here and the monocyte cells here on the top there. And the dendritic cells are presenting this to the TCR cells, TCR receptors on the T cell. The T cell then becomes activated, which then activates the B cell. The B cell, once it is activated, then transforms into a plasma cell, which then releases the immunoglobulin D, which then have got specific affinity for the basophils. And once they activate the basophils under the influence of interleukin-3 is able to transcend or move towards the site of inflammation, where it produces a number of peptidial factors and cytokines, which further initiate activation of the B cells and also initiate the transient movement of a number of immunological cells, such as the neutrophils and other cells, to the site of infection to ensure that this infection is cleared. Are there any questions? Are there any questions on the activity of the immunoglobulins, and in this particular case, immunoglobulin D. So already here, we are tackling and slowly handling the collaboration of the innate and the adaptive. As you can see, we've got the basophils. These are cells of the innate immune system. We've got the dendritic cells, which sometimes are classified either, but for now, the monocyte will classify them as being cells from the innate immune system, which are then activating the T cells through the T cell receptor. This activated T cell then activates the B cell, and the B cell initiates the production of immunoglobulins, which further activate the innate immune cells, such as the basophils. Are there any questions at this point?
The question from the system diagram.
Yes, proceed.
What you went so far, okay, for even from the same diagram, we know that the B cell can be activated in two pathways, the T cell dependent and the T cell independent. Now, when it's activated in both, let's say the pathogen invades the immune system, then the B cell is activated either, let's say it's activated in the first encounter, it's activated by the T cell independent, and then the other one is activated by a T cell independent. Are the immunoglobulin which is going to be produced, is it going to have the same affinity, or they will differ?
Okay, that's a really good question. It's good that you're looking at it from that angle. Okay, so let me put it in this way. So we've got B cell independent, we've got T cell independent activation of the B cell, and we've got T cell dependent activation of the B cell. So in this particular case, there are two scenarios that will play. Remember that this is an environment where there are a number of cells, just not only one cell. Okay, so in one case, you might actually have an antigen which has gotten or infected the particular region being detected directly by the B cell. So remember that the B cell is an APC itself. It's an antigen presenting cell. So through its BCR receptor, it is also able to directly bind naive antigens. I use the word naive, okay? It is directly able to bind naive antigens on its surface. And once it binds those naive antigens, it functions as an APC, where now the B cell then moves to go and present that antigen it has engulfed. It goes and presents it to the T cell. Remember, once it binds direct to an antigen, that is the first signal transduction of the B cell. And that initiates and increases the expression of MHC class as well as the CD40 receptors. The B cell then moves to and presents that antigen peptide. The processed antigen goes and now presents it now to the T helper cell. Okay, and once the MHC class of that B cell binds to the TCR of that particular T cell receptor, we've got now the entire process where now the B cell becomes activated in response to that presentation mechanism, and the T cell also becomes activated. Okay, so here we are talking about T cell activated mechanism via the direct antigen B cell binding. On the other hand, we can have a situation where the antigen is first detected by other phagocytic cells, such as the dendritic cell. And those dendritic cells present the antigen to the TCR or to the T cell receptor. And that T cell receptor binding to that dendritic cell, the T cell receptor becomes activated. And once it becomes activated, the T cell then will then activate the B cell. So now we have got a concept where we have got an activated T cell activating a B cell. A T cell which is activated by its interaction with another cell, such as a dendritic cell, it then initiates a process where it now activates the B cell. So upon being activated, it then activates the B cell. I don't know if you're understanding me. So we've got two ways in which where eventually the B cell is being activated. Then also now, on the other hand, the third angle, we might have an angle where we've got what we call T cell independent activation. And in T cell independent activation, this is going to happen depending on the type of antigen that is present. So if these are type of polyvalent antigens that have got multiple epitopic presentation, in which once they bind to the B cell, instead of the B cell being activated once, it is actually activated twice. From then on, the B cell begins proliferation. Okay? The B cell begins proliferation. What is important between the three activations of the B cell I've mentioned, first of all, we've got the direct antigen B cell activation. Then we've got the T cell activation of the B cell, and then we've got the T cell independent activation of the B cell. In all these processes, what is happening is that the B cell is being activated, and the type of response it initiates, it is no different whether it was antigen, whether it was T cell dependent or whether it was T cell independent. The most significant factor in what type of antibody will be produced and the strength of that or affinity of that antibody is the initial antigen itself. If it is a very strong antigen that has got multiple binding sites, okay, more of a polyvalent antigen, we're going to have a process whereby the B cell, some of the B cells are going to undergo T cell independent activation. We're going to have some of the B cell undergoing T cell dependent activation, and probably we'll even have some even undergoing another activation mechanism through other APCs within the within that particular niche or microenvironment. So the most significant factor here is the type of antigen, the concentration of that antigen. Okay. So at one point, I don't know if you've covered antibody diversity, but if you haven't, that's still fine. Okay. The type of antigen, the concentration of the antigen play very, very important was in determining the strength of the immune response in relation to the strength and affinity of the antibodies that are being produced. The strength, affinity, and type of antibodies that are being produced. Does that make sense?
Yes, sir. Thank you very much.
Awesome. Okay, it's very, very important that I love that question, because if you're able to answer that question well, it actually demonstrates your understanding of the immune system, because you need to interrogate the immune system to say, okay, what if this particular cell activates this other cell first? What happens then? Okay, you should always remember that we have got a lot of cells in this particular microenvironment, and the most determinant factor on the type of immune response, strength of immune response, is the antigen. What is the concentration of the antigen? What type of antigen is this? Is it COVID? Is it what? Okay, is it re-exposure? All those determine. Okay, is this an antigen that the body has experienced before or that has encountered before? Or is it a first-time antigen? And all these have got a determination on the time of response of all these other cells. So if it's a primary infection, having this infection for the first time, you actually expect a slow reaction from cells of the adaptive immune system. Because by the time the immune system is going to elicit a response to produce those antibodies that will come and do the further activation, we literally be at day 10. That is over a week already. And if it is a very virulent pathogen, you find that it would have already done a lot of damage. Okay, so that's why you find that the cells of the immune system also have got some form of mechanism. First of all, the first idea is, can we prevent this antigen to enter? We have got the skin. So when we start breaking down the innate immune system, you find that each of these have what you call a structured layer or arrangement of the immune system. And the initial idea of all those mucosal surfaces, skin, is let us prevent infection. Okay, if it's mucosal surfaces, you've got the IgDs and the IgAs. Their idea is to bind to these bacteria and prevent further infection through the epithelial cells within the microenvironment of the immune system. Okay, so, and this is the type of thinking I need you guys to develop for you to have an appreciation. Think of it as a system. Okay, what is happening when an antigen comes in? Okay, previously we talked of a pollen allergen. It comes in. What is happening with this particular pollen allergen? Oh, it is activating the B cell. Oh, okay, the B cell is active. What is happening now? The B cell is then activated. It interacts with the T cell. It receives its first signal. It's fully activated. It undergoes clonal expansion and proliferation and differentiation. Then produces the immunoglobulin. Is where do those immunoglobulins go? Okay, you'll find that those immunoglobulins have got an affinity for allergens, and also they've got an affinity for the FC receptor on the basophils. And once that allergen is exposed to that particular activated or armed basophil, degranulation of its contents occurs, and it releases those contents in order to, as a defense mechanism, in order to get rid of that particular allergen. However, due to the effect of those chemicals that are being produced, there's also a response there, which then causes further hypersensitivity reactions or allergic reactions in response to that particular antigen. That's how we must try to describe the immune system. Okay, and I would appreciate if as individuals we try to spend a bit of time to understand, together with a broader research to find out there, to just understand how does the B cell, how does for example, a pollen antigen initiate an immune response of the adaptive and innate immune system. And I've handled this part specifically because my next lecture, I might start talking about now the specific collaboration, and I might come back to a similar situation where I will now try to again reconnect the dots in terms of what is happening with these particular allergens, with focus on the collaborative nature of the innate and the adaptive immune system. Any other questions? Okay, there are no questions. Let's just do a summary. Okay, so in summary, antibodies are antigen binding proteins that are present on B cell membranes and are secreted by plasma cells. Membrane-bound antibodies confer antigenic specificity on B cells, as we have discussed, and also the secreted ones that are circulating within the peripheral system. These serve as effectors of the humoral immune system, connecting the complement, connecting the adaptive and the innate immune system. They are involved in antibody-dependent cell cytotoxicity. They are involved in opsonization of pathogens so that there is activation of phagocytosis by the neutrophils and the macrophages. All that is very, very significant. And also very, very important to know is that all antibodies share structural features, bind to antigens, and participate in the limited number of effector functions. And more importantly, is that when you look at the immunoglobulins, they have got particular sizes, and also here, for example, when we talk of antibody-dependent cell cytotoxicity, you can literally see which immunoglobulins are involved, okay, in complement fixation, which immunoglobulins are involved in antigen neutralization, which immunoglobulins are involved. You can literally go through this and try to understand further. Okay. Okay, ladies and gentlemen, this marks the end of my lecture today. Are there any additional questions? Going once? Any additional questions, clarifications? Okay, you've been an awesome audience. I hope it's making sense. Probably as you tackle or interact with the slides, you will find it more. Can I say you'll find it easier to understand? So, yeah. So, unless if there are any questions at this point.